Hello, this is Miss Dagenford, and we want to continue our discussion from class. We were looking at translation, and we had gotten to the point where we were looking at it as a repeating cycle. A ribosome has an mRNA strand on it, and the tRNA comes up with the appropriate anticodon, which means it has the appropriate amino acid. You make the peptide bond, and then you have translocation, which is the ribosome moving down along the mRNA strand. Now, it's going to keep doing that and keep doing that and keep doing that until it hits a termination factor. This is a stop codon. Now, the stop codon doesn't code for any amino acid, but it does allow a termination factor to come in and sit in the spot. And because there's nothing attached to it, it'll begin the process of loosening the polypeptide that was formed. And then everything disassembles. And you can see right here, this requires ATP to kind of break everything down. Now, if this is a mRNA, which, think about it, would also mean that it's a protein that's in high demand, then you can get a number of ribosomes on the same mRNA strand. And so if I were to kind of indicate that, let's say this is our mRNA strand right here, and the ribosomes, let's put them in red. And so our ribosomes, then let's say this is the beginning. Well, at the beginning, the ribosomes have only translated a couple of polypeptides. And so let's change that color. Let's say we only get a couple of amino acids to that. Well, in a polyribosome, you might have a number of ribosomes attached to it. And so we can kind of put these on all of these multiple ribosomes. And remember, this would be an mRNA that's making a protein that we want a lot of. So if this is a pancreas cell, this could be making a lot of insulin. If this is a B cell, that could be making a lot of proteins. So think about the fact that as they get down further and further on the mRNA strand, they're going to be making a lot of amino acids connected together. And so the further down you go, the more of these amino acids that they have added. And I always find it amusing that by the time we get down here, we've made our little Christmas tree. And in fact, they actually saw this under an electron microscope, this kind of shape where you saw the ribosomes further down on the mRNA strand that have made successively longer polypeptide chains because they were farther down on that mRNA. So this gets us to the question that some mRNA molecules are in high demand. So the two examples I gave, and I would like you to maybe think of other ones that we've talked about. The two examples that I gave were the pancreas cell, which is making a lot of insulin, or a B cell that's making a significant number of antibodies. And if they're churning these proteins out, then there must be a fair number of mRNA molecules to then allow them to make the proteins. So that gets us thinking about this idea of controls. How do we control what mRNA strands are made, transcribed, and then how can we then dictate whether or not they're translated or not? Now, transcriptional controls are going to make it easy for the RNA polymerase to bind. So one of the first things we see is anything that tells the RNA polymerase on a molecular level, stay here, bind here. And we're going to see this repeatedly with these transcription factors, at least on a eukaryotic level. Now, prokaryotes have a different way, and we're going to talk about that in a bit. Now, just to put something together that we've talked about before. And hopefully your mind might be thinking, what in the world is this blue thing that we're seeing? Well, this blue thing is the rough ER. And the rough ER is going to contain, we know this already, ribosomes on it. And these ribosomes, what we have mentioned is these ribosomes are making proteins, and dig back in your brain a little bit. These proteins are going to either go outside the cell, they are going to be secreted outside. Insulin is a good example. They're going to be membrane proteins. And in fact, we just did the MC1R protein in the melanin pathway in our rock pocket mouse. And then they could also be proteins that go inside another organelle, like a lysosome. But here's how it actually works. How does a ribosome, quote unquote, know that it needs to go to the rough ER? Well, this is another molecular machinery, a complex where we have a lot of things going on. And so, for instance, as a ribosome starts translating and you get this polypeptide being made, we have a few amino acids that are a signal peptide, and that turns on this idea that it's going to go to the rough ER. And so the signal peptide will interact with a signal recognition particle. Now, the signal peptide is the signal that says go to the rough ER. This is where I go. Now, the signal recognition particle is a little tugboat, if you will. And so that's what it's going to do. It's going to take it over to the rough ER, and it's going to interact with the signal receptor protein. And notice what they do. They bond together in this complex that's going to allow the polypeptide to be fed through into the rough ER. Now, the signal recognition particle disappears once everything gets started. And you'll notice in this example, this one would be a protein that's eventually going to be secreted, or it's going to go into a lysosome, because notice it's inside the rough ER. In other words, you could make this little vesicle that's going to go off to another part of the cell or outside. However, I want you to consider the possibility that maybe, just maybe, this protein, as it's being fed in, you would have the protein stay, or at least some of it stay inside the membrane. And this is indeed what we see. If you've got these transmembrane parts, these transmembrane parts are going to stay in those nonpolar areas, and then you'd have a part that stays outside and a part that stays inside. Now remember that this then flips as it gets to the membrane. So this area right here might be the extracellular domain. So this might be extracellular. It's going to go outside. Then you have the intracellular, which would stay inside. And then here, this might be the receptor portion. So remember in the rock pocket mouse example, it's those mutations that cause a different shape. And so this receptor in the normal, it might look like that, but maybe in the mutant, suddenly our receptor changes, and that changes how the whole pathway interacts with the signals. So here's kind of putting everything together with how we already know it, where it goes. We know it goes to the outside, or it stays on the membrane. Now we know how it does that. Now, I want you to be thinking about this idea of proteins that are in high demand or in low demand. And so I came up with a silly little example that might help you. If we've got a teddy bear factory and we've got a bunch of steps, and these steps, these workers are one worker is going to make the head and body of the teddy bear. Another worker might make the arms. Another worker might make the legs. And another worker might put the stuffing in the teddy bear. And then suddenly we have a completed teddy bear. Well, if since this is Christmas time, if nobody is ordering teddy bears, then the last thing you want to do is waste your supplies. If people are ordering Legos, and then you don't want to make teddy bears. So you really want to shut down this assembly line, and you really don't want to shut down the assembly line way down here at the stuffing level, correct? I mean, you're going to waste a lot of energy and time of these other workers if they are waiting until the person that does the stuffing to stop. So what we are going to be looking at are all sorts of ways to control transcription primarily, but also translation, because we really don't want to wait until translation to work it. The other thing that you're going to see is we really want to tell the entire assembly line to shut down. We don't want only one person working and everybody else shutting down. So how do we get one supervisor to basically say to everybody, all right, guys, we're not making these things. And that's what we're going to be looking at next. So hope that gives some closure to the process of translation in general, and some of the interesting possibilities of it. We'll talk about these when we get back on Tuesday, and have a great rest of your weekend.